Hello everyone, my name is Eliana Kim, and today I will be speaking about CRISPR-Cas9 and its potential as genetic therapy for patients with cystic fibrosis. So, a little about me. I am a rising junior at the School of Advanced Studies at North Hollywood High. I'm an aspiring pediatrician or neonatologist with a strong interest in biotechnology as well. At school, I'm the Public Relations Chair of the Associated Student Body, as well as the founder of the Sending Sunshine chapter, which works to reduce senior loneliness with handwritten cards. I am also the Vice President of the UNICEF USA chapter at North Hollywood, helping fundraise for children in need. Additionally, I am a regular volunteer at the Koreatown Youth Community Center. I am a pianist and a competitive archer on a regional, state, and national level. So, what is CRISPR-Cas9? Discovered in 2012, CRISPR is a groundbreaking genetic engineering tool that acts as a programmable molecular scissor for precise gene editing by cutting and modifying specific DNA sequences. This tool's versatility spans biomedical research, agriculture, and the treatment of genetic diseases, making it a significant focus of global scientific efforts. One such genetic disease is cystic fibrosis, or commonly referred to as CF, a severe disorder caused by mutations in the CFTR gene, leading to thick mucus production that impairs respiratory and digestive functions. It is inherited in an autosomal recessive manner and is associated with specific genetic mutations, including nonsense mutations that result in non-functional proteins. An estimated 105,000 people live with diagnosed CF around the world, but approximately 1 in 30 people are carriers of this genetic disease. CF is a lifelong condition requiring extensive healthcare resources. Its management poses significant challenges, particularly in low-resource settings. CRISPR-Cas9 offers a promising approach to correct genetic mutations due to its precision and cost-effectiveness. Using organoids, lab-grown cell masses mimicking organs, researchers can study and potentially treat CF outside the patient's body. This research not only has the potential to improve the quality of life for CF patients globally, but also advance our understanding of other genetic disorders. Addressing such genetic diseases with innovative technologies like CRISPR-Cas9 is crucial for reducing healthcare disparities and improving global health outcomes. So, how exactly does this work? In this study, scientists worked with mini-guts from individuals with cystic fibrosis. The researchers developed a culture system to grow small 3D intestinal organoids from stem cells, which can become different types of cells. They targeted and mutated the APC gene, a gene that usually controls cell growth, causing the cells to grow abnormally. Sequencing confirmed the mutations occurred as intended. Targeted or corrected is actually a very general term for this type of process because the way CRISPR functions involves designing specific guide RNAs to target the mutation, then cutting the DNA at the mutation site with a Cas9 enzyme, and then using a donor DNA template to replace the faulty sequence through the cell's natural repair mechanisms. The scientists then used CRISPR-Cas9 to insert a healthy CFTR gene into the patient-derived organoids. They included a silent mutation and a puromycin resistance marker to identify successfully edited cells. After selecting these cells, sequencing confirmed the precise insertion of the healthy gene, and RT-PCR analysis verified its expression. To ensure the CRISPR-Cas9 edits were accurate, they analyzed potential off-target sites and found minimal unintended changes. When they tested the corrected organoids with forskolin, they swelled, indicating restored CFTR function. For clarification, on a molecular level, forskolin raises the intracellular CAMP levels, which then activates the CFTR protein, causing it to allow chloride ions to flow through the cell membrane. Quantification showed a significant increase in surface area compared to uncorrected organoids. This picture here shows that forskolin-induced swelling in the corrected organoids was sensitive to chemical inhibition of CFTR, indicating the restored functionality of the corrected allele. Figure A on the right shows the normalized area percentage of organoids over 10 minutes under different treatments, and Figure B shows images of the normal, or wild type, versus the mutated defective CFTR gene organoids at 0, 5, and 10 minutes to compare their swelling. In conclusion, with the precise editing of CFTR mutations using CRISPR-Cas9, there was a reflected enhancement in chloride ion transport, reduced mucus production, and overall improved cellular and physiological characteristics associated with cystic fibrosis. The graphs displayed in this conclusion show the effects of this process on the small and large intestinal organoids, which were the original mini-guts derived from CF patients. Overall, this research demonstrates that CRISPR-Cas9 can correct the CFTR gene in CF patient cells, restoring its function with high precision and minimal off-target effects. These findings are promising for developing new technologies and treatments for CF and other genetic disorders, highlighting the potential and safety of CRISPR-Cas9 technology. Of course, there is far more research to be able to be done concerning the future advancements of CRISPR-Cas9. However, the combination of HR-based gene correction strategies with organoid culture technology provides a potential strategy for future gene therapy in patients with single-gene hereditary defects. Appropriate acknowledgements go to the researchers that are associated with this groundbreaking research: organoid architect Hans Clevers, co-discoverers of the CRISPR-Cas9 technology Emmanuel Charpentier and Jennifer Doudna, and several others noted on the right. Here are a few additional resources. If you would like to contact me, attached is my email and my LinkedIn. Thank you so much for listening.